our traditional human interpretable features do an excellent job at characterizing what's going on in the tumor microenvironments. And what we can get from nuclei features is basically additional orthogonal readouts of what's actually going on, both at the prognostic level as well as at the molecular level. And so the goal of the project was to build a segmentation tool so that we could characterize the shape of nuclei and characterize their morphology and be able to use that to predict these prognostic values. Some of the features that we were trying to predict were whole genome doubling, also known as WGD, homologous recombination deficiency, which is HRD. And then in prostate cancer, we looked at the Gleason grade, specifically differentiating Gleason grade 8 through 10 from the less severe Gleason grade classifications. And in breast cancer, we also looked at HER2 positivity status. This was a three-step approach where the first step was to build an object detection algorithm. And so what this is basically building bounding boxes around the nuclei. And then we applied a segmentation tool where we can actually get a pixel by pixel classification of whether or not that pixel contains a nuclei. We were able to extract what we call nuclei human interpretable features, which characterizes the mean and standard deviation of various features across all cell types of a particular class. And what some of these features is the circularity of the nucleus, the eccentricity of the nucleus, which is basically how ovular it looks, makes it able to squeeze through basement membranes into the vasculature tissue to basically spread to other parts of the body. We also looked at the coloring of the nucleus, the staining intensity, and the texture of the nucleus, which is basically how rigid the nucleus is. In each of the breast, prostate, and lung cancer cohorts, we first ran a logistic regression model with an elastic net penalty. So features that were uninformative to the prediction would be dropped out, and any features that were collinear with each other would also be dropped out, only keeping one of those features. And when we looked at whole genome doubling across breast, prostate, and lung adenocarcinomas, what we saw was that the eccentricity, the major axis length, and the minor axis length of the nucleus were associated with increased whole genome doubling in both breast and lung adenocarcinomas. When we looked at prostate cancer, the features differed slightly. The major axis length was still associated or predictive of the whole genome doubling within prostate cancer, but the grayscale intensity as well as the coloring of the nuclei were the other two more informative features. It's important that we can make these predictions from H&E whole slide images. So one, if we can be able to predict homologous recombination deficiency through our features, including our nuclei features, then we can circumvent the need to basically do the genomic sequencing, which at the clinical level costs about $4,000 to $5,000 per patient. And so that really adds up. We can gain additional insight into what is going on with the tumors, potentially if the patient is going through relapse or they are going to have worse prognostic outcome than we originally thought based on their original pathological biopsy or their DNA sequencing